Welcome to Laylasceincehub. Today we are diving into microscopes, one of the most important tools in biology. But have you ever wondered why we need different types? Why isn't one microscope enough to see everything? Well, today we're breaking down the different types of microscopes, their advantages and disadvantages, and what you need to know for A-Level Biology. Let's get started. Let's start with the one you've probably used before. Light microscopes, also called optical microscopes. These use visible light and glass lenses, making them cheap, portable, and easy to use. One of their biggest advantages is that they can be used to study whole organisms and living cells. Plus, they allow us to use stains which help add contrast to see structures more clearly. However, there are some disadvantages. They have a low resolution and magnification, so you can't see organelles in detail. So what exactly can we see with an optical microscope? Here are some examples. Living organisms, like amoebas in water. Smears, such as blood or human cheek cells. Thin sections of tissue. Now, what if we want to see more detail beyond what a light microscope can show us? That's where electron microscopes come in. Unlike light microscopes, electron microscopes uses a beam of electrons, which are focused onto a screen or photographic plate via magnets. This allows them to produce images with much higher magnification and resolution, revealing details in cells that light microscopes simply can't show. There are two types of electron microscopes, the transmission and scanning electron microscope. Unlike light microscopes, these are more expensive and require expert skill to use. First, we have the transmission electron microscope. This is the one we use when we want to see inside cells. For example, to study organelles like mitochondria or ribosomes. It has a higher resolution and magnification than light or scanning electron microscopes, and it works by passing electrons through a dead, thin specimen to produce a black and white 2D image. Now, what if we want to see the surface of a specimen instead of its internal structures? That's where the scanning electron microscope comes in. This is perfect for looking at surface details, for example, the outer structure of cells, or even tiny organisms like viruses. It works by electrons bouncing off the surface of the dead specimen to produce a 3D image which false color can be added on afterwards. But what if we wanted to study living cells in action? That's where our last type of microscope comes in. Unlike electron microscopes, laser scanning confocal microscopes can be used on living specimens. They use fluorescent dyes and a laser beam to scan at different depths, creating high-contrast 3D images. Some advantages of this microscope are that it has a high resolution. It can be used on living specimens. It creates a 3D colored image, and it has a depth selectivity, so greater contrast can be achieved. However, it is expensive and requires fluorescent dyes. So what can it view? Cellular and subcellular structures, biological specimens, and living cells, making it perfect for tracking real-time cellular processes. Exam tip. You might be asked to compare microscopes, so remember to focus on resolution and magnification differences, whether living or dead samples can be used, and the type of image which microscope produces. Knowing these differences can help you ace those comparison questions.